Okay, here is DNA, the genetic material. This is the start of chapter 16, which deals mostly with some very historical experiments on how we learned about DNA. So, the march to understanding that DNA is the genetic material. We have quite a few people that played a role in this, and so we're going to go through each of these. So let's start first of all with T.H. Morgan. We learned about him in genetics. He worked with Drosophila, fruit flies. He associated the phenotype with a specific chromosome. Okay, the white-eyed male had a specific X chromosome. So once he figured that out, then we know that genes are on chromosomes. So he actually figured that out with the investigation that we studied earlier with X-linked traits. So Morgan's conclusions, genes are on chromosomes. But is it the protein or the DNA of the chromosomes that are on the genes? So if you remember down here, here's the chromosome. It's actually wound around proteins called histones. So we have a spacer histone and we have a central histone. So there's, chromosomes are not just DNA, they're actually proteins as well. So he didn't know whether it was the protein or the DNA. So initially proteins were thought to be the genetic material. Well, think about what proteins can do. The fact that they're enzymes and the fact that they can do so much work on throughout our bodies. And we knew this at the time, that it was fairly logical to think that proteins were the one that were doing the acting. So let's see how they designed an experiment here. The transforming principle. This is Frederick Griffith. He used a bacteria called Streptococcus pneumonia and was working to find the cure for pneumonia. Harmless live bacteria, we're going to call it rough, mixed with heat-killed pathogenic bacteria, and we'll call this one smooth, causes the fetal disease in mice. So remember, pathogenic here means that it's going to have the ability to kill the mice. So a substance is passed from the dead bacteria to the live bacteria to change their phenotype. This is the transforming principle. We're actually going to do a transformation with some bacteria in a lab as we're learning this. So here is the actual experiment called, again, the transforming principle. So if you take a live pathogenic strain of bacteria, the mice die. If you take a live non-pathogenic strain of bacteria, the mice live. If you heat kill, in other words, if you boil the pathogenic bacteria, then the mice will live. So something is denaturing the genetic material there. But if you mix the heat-killed pathogenic and the non-pathogenic bacteria, then there's something about changing that DNA and allowing it to be incorporated into the non-pathogenic bacteria so that it kills the mice. So something is being transferred from the non-pathogenic bacteria, something is being transferred from the pathogenic bacteria to the non-pathogenic bacteria. So, transformation. There's a change in phenotype. Something in the heat-killed bacteria could still transmit disease-causing properties. So, DNA is the transforming principle. Let's see this. Purified both DNA and proteins separately from the Streptococcus pneumonia bacteria, which will transform non-pathogenic bacteria. So we're going to inject the protein into the bacteria, and it has no effect. We inject the DNA into the bacteria, and it's going to transform the harmless bacteria into the virulent bacteria. And the mice die. So now we prove that it's DNA. So let's go ahead and look again. Conclusion. The first experimental evidence that DNA was the genetic material. So let's confirm it. Let's do another experiment. Hershey and Chase here, this is the classic blender experiment. Up here is something called a bacteriophage, this sort of like robotic looking thing. It's actually a virus that infects bacteria. Inside here you have DNA. So viruses that infect bacteria are called bacteriophage. So we grew the phage virus in 2 media and then radioactively labeled with either heavy sulfur or heavy phosphorus. So we labeled the proteins with the sulfur and the DNA with the phosphorus. So why do we use these? Well, it's going to allow us to actually see the difference as we go through each of the mediums. So let's go ahead and look at this. Infect the bacteria with labeled phages. So if you look up here at the top, you have a protein coat labeled with the sulfur. If you look over here, you have DNA labeled with the phosphorus. So as we work our way down, sulfur versus phosphorus, the bacteriophages infect the bacterial cells. The DNA gets inserted into the bacterial cells. And over here we find that the sulfur and the protein is found in the medium, in other words, not in the cells, in the outside, but the phosphorus is found actually in the bacterial cells. So which radioactive marker is found in the cell? It was the phosphorus. Which molecule carries viral genetic info? Has to be DNA then. So here's the same experiment again. Just a little bit of, this is the medium that they're talking about. So they grew the bacteria in here with sulfur, and then down here they grew this one with phosphorus. Again, the phosphorus is going to be incorporated into the DNA, and over here the sulfur is incorporated into the protein coat. You blend both of them after you see what happens, and then this is the difference between where do you find things. So they put something in a centrifuge, which is essentially spinning things around really, really fast to create a gravitational force so that the heavier things end up at the bottom of the test tube and the lighter things are at the top. So the pellet's going to contain the cells, and here this part, the pink part, will contain the medium, which for this is the pink sulfur labeled protein. Down here you have the DNA labeled cells, that because the DNA was injected into the cells where the protein is not injected into the cells. So the radioactive phage in the bacteria in a blender. So the sulfur phage, radioactive protein stayed in the supernatant. That's the liquid that's not the pellet after it's been centrifuged. The liquid is called a supernatant. So therefore the viral protein did not enter bacteria. And here you have the phosphorus phage is the radioactively DNA stayed in the pellet where the cells were. Therefore, viral DNA did enter the bacteria, confirming DNA is the transforming factor. So, let's move on with the history of DNA. Chargaff here, DNA composition, we have something called Chargaff's rules. That it varies from species to species, but all 4 bases not in the equal quantity. So bases present in the characteristic ratio. So in humans, 30% A, roughly 30% T. Guanine is about 20%, and cytosine is also about 20%. So we now learn that, of course, A has to equal T and C has to equal G. So here we have our DNA base pairs. Interesting.